Okay, in the interest of time, we'll just get started. So good afternoon, everyone. My name is Dr. Faye McLeod, and on behalf of the Epilepsy Research Institute, I'm delighted to welcome you to our symposium today on radically advancing research into epilepsy. Epilepsy is one of the most prevalent serious neurological conditions, with 1 in 100 people in the UK equating to just over 600,000 individuals and 50 million people worldwide living with the condition. With epilepsy accounting for 3 deaths per day and 1% of all NHS costs annually, clearly urgent action is required. Over the past decade, we have seen research deliver significant advances in the treatment and care of people with epilepsy, including treatments and surgical techniques for specific types of epilepsies, devices that can modulate brain activity to reduce seizures, advanced neuroimaging to identify brain abnormalities, and around 1,000 protein-coding genes have been linked to epilepsy. However, there is still a long way to go, and many more breakthroughs are needed. So this is where the Epilepsy Research Institute comes in. We serve as the central hub for epilepsy research for the epilepsy research community. Our mission is to radically advance research into the causes, prevention, and treatment of epilepsy and associated condition. The Institute aims to strengthen the epilepsy research ecosystem by driving strategic investment and developing partnerships and collaborations between academia, the NHS, industry, funders, patient groups, and people affected by epilepsy. Central to all what we do is the culture of advocating and actioning the research priorities of people affected by epilepsy. Our strategy is underpinned by 6 key research themes, each with a task force comprising pre-eminent scientists and clinicians together with future research leaders. These include neurodevelopment, advanced therapeutics and disease modification, mortality, risk and morbidity, reproduction and hormones, capacity building and enabling technologies. Ultimately, we want to build an infrastructure that will attract the best researchers, foster an ambitious and inclusive culture of collaboration, and enable research into epilepsy to flourish. Today, we are incredibly proud to champion 4 early career researchers at various early career stages from across the UK, working in epilepsy and associated conditions. Although the research presented in the symposium will have an epilepsy focus, there is a huge relevance for other neurological conditions and general neuroscience research. So we hope to facilitate network and potential collaborations across the research areas and conditions too. In summary, we firmly believe that a life free from epilepsy is possible, but only through research. With that, I'll now hand over to the chairs of the session, Priya Menguel and Amy Richardson, who will formally introduce our speakers for today. But please do come and chat to us more at the stand 33 to find out more about our upcoming events, including our inaugural research conference that's happening in May on the 22nd to 23rd of May. And essentially that's going to be, we've got some tickets that are still available over the next week. So please do come and chat to us about that. So do check it out. And also we have our CEO here in the audience, Rosemary Partington, so please come and chat to her at the end of the session if you've got interesting collaborations, or at the stand with one of us and Rosemary over the next couple of days. So I'll now hand that over. Thank you very much.
So, hello, I'm going to introduce the first speaker. My name is Dr. Jose Prius, and I'm going to introduce Amy Richardson. Amy Richardson is an Epilepsy Research Institute Emerging Leader Fellow at the Institute of Neurology, University College London. Her broad research expertise focuses on understanding the mechanisms underlying seizures and developing genetic therapies for epilepsy. In her Emerging Leader Fellowship, Dr. Richardson's research proposes a new gene therapy treatment to try and target not only seizures, but impaired memory to improve the overall lives of people with epilepsy. More recently, her personal interest expanded into the causes and treatments of catamenial epilepsy, in addition to the mechanisms of how reproductive hormones influence seizures in both males and females.
Thank you, Prius, and thank you very much to the Epilepsy Research Institute for organising this and for inviting me to come and talk today. So, as Priya said, I'm going to talk to you today about a new gene therapy that I've been developing at University College London that is based on this protein called KCC2. So around 1/3 of people with epilepsy don't achieve seizure freedom with current medications. And for this group of people, sometimes the only option left is to surgically remove the area of the brain that's involved in the epilepsy and that generates the epilepsy. And even then, sometimes this isn't available as an option if the eloquent cortex is affected. And this really highlights the need for new therapies to treat this group of people with epilepsy that don't respond to current medications. Now, we could develop new drugs, but the problem with drugs is that they often act by dampening activity in the entire brain. And this means that they can often come with nasty side effects, such as problems with mood, drowsiness, and problems with memory, not to mention other effects on the rest of the body and the developing fetus. And so gene therapy may offer an alternative precision treatment that we can use to treat only the area of the brain that is involved in generating the seizures. And we hope that by doing this, we can reduce side effects on the rest of the brain. So what is gene therapy and how can we use it in epilepsy? So gene therapy involves inserting genetic material, usually in the form of a therapeutic transgene, into the brain, often using things like viral vectors, such as the adeno-associated virus. And usually we do this to try and increase the amount of a particular protein in the brain. And as I said, gene therapy can potentially offer a treatment that has fewer side effects, not only because we can just put it into a certain area of the brain, but we can also bias the expression of protein to particular cell types by using different AAV serotypes, which have different tropisms for different cells, and by using cell-specific promoters to ensure that our protein of interest is only expressed in certain cell types. Now, often when we think of gene therapy, we think of using this to supplement a disease gene with a healthy version to try and correct some kind of monogenetic disorder, in this case a monogenetic epilepsy. But we can also use gene therapy to change neuronal properties. And in epilepsy, which we may think of as a disorder of hyperactivity, we often want to try and decrease neuronal excitation or increase neuronal inhibition. And the gene therapy that I'm going to talk about aims to increase the natural inhibition in the brain. So we know that seizures can occur and spread across the brain when there's some kind of breakdown in the brain's inhibitory system. And there are kind of a few main hypotheses on how this breakdown of inhibition could occur. The first of those is that a buildup of extracellular potassium in the external environment can lead to depolarization of neurons and hyperexcitability. The second is that depolarization block within interneurons can prevent them from firing, prevent the buildup of GABA or reduce the amount of GABA in the system and decrease inhibition. And the third is that a buildup of chloride within principal neurons in the brain can lead to decreased inhibitory drive. And it's this third hypothesis that I'm going to be focusing on today. So in a normal adult healthy brain, when we activate our GABAergic interneurons, GABA binds to the GABA-A receptor, and usually we see small amounts of chloride entering the neuron. And this is due to a low internal chloride concentration in the cell. And this chloride moving into the neuron hyperpolarizes the cell. Now, when we get activation of GABA receptors at the same time as a depolarization of the neuron, which might happen at the start of a seizure when there's lots of activity going on, we actually increase the gradient for chloride entry into the cell. So we increase the driving force for chloride coming in. And that means that when we activate GABA receptors in this situation, we get much more chloride coming into the cell, and that can build and build and build inside the cell. And when it does that, it can disrupt the chloride gradient within the neuron. And that means that when you subsequently activate GABA-A receptors with your GABAergic interneurons, you actually get much less chloride coming into the cell, so decreased inhibitory drive. Or in extreme cases, you can get chloride moving out of the cell, which means that the GABAergic interneuron input actually can become depolarizing and, in extreme cases, excitatory. And so our kind of hypothesis or our idea is that if we can prevent this chloride loading from happening, then potentially we can prevent seizures from occurring and spreading across the brain. So the chloride gradient is set by two membrane-bound cotransporters. We have KCC2, which is the potassium chloride cotransporter 2, and NKCC1, which is the sodium potassium chloride cotransporter 1. And in immature neurons in the developing brain, we have lots of this NKCC1 and not much of KCC2. So NKCC1 is pumping chloride into neurons, and because we have so much of this and so little of KCC2, which is removing chloride from neurons, we actually have a high internal chloride in these neurons. And that means that GABAergic transmission in the developing brain can be depolarizing and excitatory. Now, during maturation, you get a switch in the expression of these transporters such that we now have lots of KCC2 and not much NKCC1, and we have a low internal chloride concentration, which means that our GABA in adult brains, as I've just said, is hyperpolarizing. But in diseases such as epilepsy, stroke, traumatic brain injury, and many others, it's currently emerging, you get a reversion into this sort of developing-like state where you get decreases in KCC2 expression and increases in NKCC1 expression. And this means that that you can kind of exacerbate that effect of chloride loading, because now you have even less KCC2 in the system to remove chloride from neurons. And so if we can prevent this reversion to this developing-like state by overexpressing KCC2, then maybe we can prevent seizures. So we designed I designed a bunch of different KCC2 mutants. So one thing we also know is that wild-type KCC2, not only is it decreased in epilepsy, but also there is reduced membrane expression in different models of epilepsy. And so I aim to try and introduce mutations into our KCC2 construct, which would stabilize our KCC2 protein on the membrane. And so I designed a number of these different constructs and tested how good they were at removing chloride from neurons. And so I did this by measuring the reversal potential for GABA. And basically, what I want you to take away from this is that the more negative the reversal potential for GABA, the better the construct is at removing chloride from neurons. And so you can see here in green, we have EKCC2, which was a construct with 3 different mutations that stabilize KCC2 on the membrane. And we can see that it has hyperpolarized eGABA the most, which we then took to mean that this was the most effective at removing chloride from neurons. And so I used this EKCC2, which stands for enhanced KCC2, packaged it into an adeno-associated virus, and I used this to try and increase my levels of KCC2 in the brain. And so the first thing we wanted to do was to check to see if our construct could actually increase the levels of KCC2 in the brain. And so we designed some experiments to look for the expression. And you can see in our injected hippocampus, we have lots of staining. So there's lots of green in here for our flag epitope, which is just a little tag on the end of our KCC2 that we can use to detect whether it's there or not. And in our non-injected side, we have no flag present. And so we know that we can deliver our gene therapy effectively to the brain. And then we wanted to look at protein expression levels. And we can see that in animals injected with GFP virus, which we used as a control virus, we have standard levels of KCC2, which are much increased in animals injected with our enhanced KCC2 gene therapy, which is great. And the next test was to see whether this increase in KCC2 was actually upheld in a chronic model of epilepsy, where we know that KCC2 levels might be decreased. And we showed that, indeed, the KCC2 levels are still increased in chronic epilepsy. And so we know that our KCC2 is not being taken from the membrane and is not being decreased. And the next thing we wanted to do was to see if our enhanced KCC2 therapy was effective at reducing seizures. And so to do this, we used an in vitro model of seizures where we used a low magnesium solution coupled with electric stimulation of the entorhinal cortex. And this develops these sort of seizure-like events. We won't call them seizures, but they're seizure-like events. So this is just an example in a slice that was treated with a GFP control virus and our EKCC2 therapy. And we found that in all of the slices injected with our therapy, the seizure duration was massively reduced, and the number of spikes per seizure was also reduced, and the interspike interval was not quite significantly increased, but it was increased. And so not only are we able to deliver our virus to the correct area, but it's also functional in reducing seizures or seizure-like events. So next we wanted to test this in an in vivo model of seizures. So we chose an acute model to start with, and we took mice of around 2 months of age and injected bilateral hippocampus with either our therapy or a GFP control. And we left the mice for 3 weeks for the AAV to express. And then we conducted some acute status epilepticus experiments where you inject repeated amounts of subthreshold canic acid, which is just a proconvulsant, and induces seizures. And then you behaviorally score how severe the seizures are that the mice are having, using this Racine scale that we have here. And we found that when we did this, that our mice injected with the therapy overall scored much lower on the Racine scale compared to those with GFP. So all animals injected with the control virus, apart from one, went into the most severe stage of status epilepticus, whereas those injected with our therapy were much more the lower end of the scale. And so not only can we prove that this works in vitro, in slices, but also it seems to be working in vivo. And now we're currently testing this to see whether it's effective in spontaneous seizures in a chronic model of temporal lobe epilepsy. And so, in summary, gene therapy can offer a targeted treatment approach for refractory epilepsy. I hopefully convinced you that chloride loading can lead to a breakdown of inhibitory strength, and by correcting this, by using the overexpression of KCC2, we can reduce seizure severity and increase seizure threshold. And potentially this may offer a promising new approach to treat refractory epilepsy. And with that, I just want to say thank you to not only the Epilepsy Research Institute for funding me, but also a number of students in my lab that have contributed to this work, as well as Kate, who is now a postdoc in my lab. And thank you very much for listening.
So thank you for the talk. And now if anyone has questions, is the moment. You can raise your hands and I will, well.
Hi, lovely presentation. Just a quick question. Did you try to look at differential expression of KCC2? So look at a gene dosage effect. Is it all or none or?
Yeah, no, so I haven't. It's a very good question. So we've done this with some other gene therapies in development in the department, actually. And what we find is that, so we tested one that was between a 10 to 10 dose and a 10 to the 13 dose of virus. And we found that actually the protein expression within that doesn't change very much. It seems to be that it increases and then it sort of hits a ceiling. I haven't tried that with my KCC2, but it's something that I should do and see if we do get a dose response.
And just a cheeky second question. So KCC2 is the right isoform. Are there other isoforms KCC which are worth looking at?
Yeah, so there's a KCC2A and a B, and what we're targeting is the B isoforms, which is mostly expressed after postnatally. So KCC2A is expressed a lot prenatally and during development, but then the expression drops off a lot in developed, mature brains. So we're mostly targeting B.
Okay, thank you.
Thank you. Very, very interesting approach. Thank you very much. I don't know anything about adenoviruses. Once you inject them and you induce protein expression, how long the protein expression lasts? I mean, from a therapeutic point of view, do you envisage multiple injection or one is enough? That's one question. And at the beginning of the talk, you mentioned the possibility of also using not just different viruses, but also cell-specific promoters. But in this case, you didn't use, or you didn't talk about it.
Yeah, okay.
Thank you.
So second question I'm going to come to first because it's the easier one. So I did use a cell-specific promoter for this. So I was targeting only pyramidal neurons because we don't want to increase inhibition of inhibitory interneurons and then disinhibit our pyramidal neurons. So I was using a cell-specific promoter to only target excitatory neurons for this. The first question, AAV. So the first question, we expect this to be a sort of one-shot treatment. So AAVs have been shown to stay within the brain for at least 10 to 15 years in non-human primates and in humans. So there were some studies on Parkinson's from 20 years ago, I think, where pieces of brain were taken post-mortem from patients who had been injected with a Parkinson's gene therapy, and it showed that the increased amount of protein was still present after 15 years. Well, I think that was 10 years, and then in the non-human primates, it was there after 15 years. So we know that it's a long-term treatment. There haven't been studies done long enough yet to say whether it is a lifetime treatment. But we expect that the AAV sticks around for a very long time. So it exists as a sort of episome within the cell. So it doesn't integrate into the DNA like lentiviruses.
Hello, thank you for your presentation. And I have a question that, are you planning to validate your research on a different epilepsy model, for example, pilocarpine or anything else? Because from my own experience, I know that results can vary extremely depending on the model used in experiment.
Yeah, so I'm currently testing in a model of temporal lobe epilepsy on spontaneous seizures, but also Kate is going to be working on testing this in a model of focal cortical dysplasia, so a genetic model of epilepsy. And we know that in a lot of different models of epilepsy, the KCC2 is actually reduced, so we would like to test some more as well. We know specifically tuberous sclerosis has this decrease in KCC2, so we'd be interested in testing that too.
Thank you.
Thank you, Amy. I have many questions, but let's maybe two quick. Have you tried the experiment the other way around, which means injecting the virus after the induction of the seizures to see how neurons that are epileptic already express and how they respond?
Yeah, so that is the experiments that I'm currently running. So we inject animals with canic acid, wait 2 weeks for them to develop chronic epilepsy, spontaneous seizures, and then record in vivo baselines for 2 weeks, inject our gene therapy, and then record for another 2 weeks after a 2-week waiting period. So you have a before and after when animals are already spontaneously seizing. So it's more reminiscent of an actual epilepsy.
Exciting. Looking forward to see. The second, do you know what happened with interneurons in this model? Are they affected also? Because you are then expecting that they function normally, let's say.
Yeah, so we know in this model that especially PV interneurons are decreased. There is some decrease in somatostatin, but not as much. So we do know that there is interneuron loss, and it's a feature of this model. But we hope that by using KCC2 in pyramidal neurons, we can strengthen the inhibition that's still there with the remaining interneurons.
We have time for one more question, or a couple of more questions. So, yeah.
Thank you very much. I was thinking about some of the other diseases you mentioned before where it's reduced traumatic brain injury or stroke. There are also diseases where spontaneous seizures can arise later on. Is there any kind of hint from the literature that the drop in expression level might be a predictor of whether seizures will start in those other diseases or not?
Yeah, it's a really good question. I'm actually not sure. I think, so a lot of the expression studies have been from human patients, post-mortem, or people having epilepsy surgery. And so it's difficult to tell whether it's a cause or consequence of the seizures or the disease. But no, it's a really interesting question that I'm not sure of the answer, whether it's
see if the pathologist can get the clinical notes to see whether they had seizures or not.
Yeah.
Anyone else? Questions? Hands up? No? Okay, so then we can...
Thank you very much. Okay, so I am now going to present Prius. So Dr. Jose Prius Mengual, who prefers to be called Prius, is an Epilepsy Research Institute and NATA Emerging Leader Fellow at the Kavli Institute for Nanoscience Discovery, University of Oxford. He completed his PhD at the University Pablo de Olavide before taking on a postdoctoral research scientist position at the University of Oxford in the lab of Vladislav Vyazovsky to investigate the role that sleep plays in cognitive function and how disrupted sleep has been linked to a broad range of neurological diseases. So in his current Emerging Leader Fellowship, he aims to characterize the relationship between epilepsy, sleep, and ER stress. And he uses a novel conditional knockout animal model to reveal the molecular mechanisms linking synaptic function, neural excitability, and unfolded protein response, and investigates whether modulation of ER stress pathways linked to sleep can prevent seizures. So, welcome, Prius.
Thank you very much. So, okay. I'm going to talk about sleep, epilepsy, and endoplasmic reticulum stress. That is a very specific kind of cellular stress. So we are going to introduce those concepts and how they relate to each other so we can understand why I'm doing what I'm doing. So what is epilepsy? Epilepsy is an umbrella term that englobes neurological disorders characterized by enduring predisposition to generate epileptic seizures. It could have multiple origins, could be genetic, could be infection, could be immune, or could be even unknown origins. But in the clinic, they are classified by focal, generalized, or unknown onset of epilepsy. It's focal when the origin of the epilepsy is a specific region of the brain, and we know where it happens. It's generalized when there is not a specific origin in the brain for it. And unknown when the clinical test is unable to tell us if there is a focal point of origin or not. For this talk, the important thing is the electroencephalogram features or the EEG features, or the electrical activity of the brain, specifically the interictal spikes and the ictal activity. The interictal spikes are a small burst of activity that happen in the brain and normally do not correlate with behavioral manifestations. And the ictal activity is what normally originates the seizures in the brain. Now, when we refer to sleep, sleep is present in animals studied to date, from the small hydra to the big whale, and is characterized by a series of changes in behavior, in molecular changes, and electrical activity changes that allow us to classify different vigilance states that are normally grouped between sleep or wake, and in animals with more complex nervous systems, these sleep stages could be non-rapid eye movement, or non-REM, and rapid eye movement, or REM. It's important to notice that the specific features defined in each vigilance state change between species due to the need of each animal to adapt to its environment. In the case of my research, I use mouse model, and mouse is a very good example that is very similar to humans. It's easy to work with, and it has many similarities. Finally, what is the endoplasmic reticulum stress? The endoplasmic reticulum is an organelle that is in most cells, and is especially important in neurotransmission because it's present in pre- and postsynaptic terminals and help with the fine regulation of neurotransmitters, vesicles, etc. When the endoplasmic reticulum is suffering an overload of proteins, of protein synthesis, we have an increase in the amount of misfolded proteins in the lumen of it, and that will trigger this pathway that is called unfolded protein response. That is the main molecular target for my therapy. It always starts the same way. The small chaperone protein BIP will be associated with 3 different membrane receptors that are looking to the lumen of the endoplasmic reticulum. And when we have a higher enough concentration of misfolded proteins, the affinity of the BIP protein will change and will dissociate from these membrane receptors. Well, will trigger the pathways. The important message here is that this unfolded protein response has an early stage that tries to restore protein homeostasis. Is kind of the good part. And it will try to do so, blocking protein translation, trying to promote the generation of more chaperones, or inducing higher activity in protein degradation complexes. If they can't resolve the stress, it will trigger a late response that is a pro-apoptotic response. And now that we have more or less pictured these 3 concepts, how do they relate to each other? So I'm going to start speaking a bit about sleep and near stress. So this is data that I got a year ago or so that reproduces what was done in the early 2000s, in which sleep deprivation was sufficient to induce an increase of ER stress genes. We see here that we are talking about BIP, this is a small chaperone that is the initiator of all these pathways, and also XBP1, that is a small RNA molecule that, when active, goes to the nucleus and starts triggering different signals, like generation of more chaperones or generation of other early gene actuators. Late genes like ATF4 that are more related to the apoptotic pathways of the unfolded protein response are still not active at 4 hours of sleep deprivation. And that's kind of good because we know that sleep deprivation triggers ER stress, but we don't have a detrimental situation. Now, what is the relationship between epilepsy and ER stress? So a paper from a couple of years ago was working with mutile mice for NMDA receptors. And they tried to improve the mice inducing more synthesis of the BIP protein. That was sufficient to recover, to come back to normal wild-type amount of NMDA currents. On the other hand, overexpression of XBP1, this small RNA that I mentioned just before, creates seizures in animals when it's continuously expressed. So the main idea here is that manipulating the ER stress gene will modify electrical activity in the brain. And also in humans, 1 year before these 2 papers, it was published that when you take control brain from people that do not have epilepsy, and you test it with brains of people suffering epilepsy, there is an overexpression in these genes, in these ER stress genes. And this happened in different groups. ELA1 refers to the group of people who are seizure-free under medication, and 2 and 3 are people who still have seizures despite having medication. Yeah. And now, how is sleep related to epilepsy? For the context of this talk, I'm going to talk about sleep in rodents. And the important thing to notice is rodents have 1 wake, 1 non-REM, and 1 REM states that are characterized, but the wake state would be a slow amplitude of the EEG, but high amplitude of the electromyogram, that is the muscle activity. The EEG during non-REM would be high and a small EMG, activity, and the REM phase will be characterized by theta activity with very low EMG activity, very low muscle activity. The important thing here is that the high amplitude of the EEGs is because they are naturally occurring slow waves during non-REM. These slow waves also follow a dynamic. This dynamic is that they are higher in they are more frequent and higher in amplitude at the beginning of sleep, and the longer we sleep, the smaller they are. And this is not happening in people suffering epilepsy. In people suffering focal epilepsy, for example, we can see that around the focal origin of epilepsy, we have more slow wave with higher amplitude, and this happens even during wake. And this renormalization of a slow wave over time is not happening either. So we know that epilepsy is changing normal long-term sleep EEG activity in the brain. So the challenge would be supporting long-term recovery of normal brain function and not just seizure suppression when we come to new development of new therapies. And for that, I'm going to start speaking about my project. So my goal is to generate a treatment against epilepsy that is not disease-specific. And for that, I'm going to use this animal model that is called TBC1D24. That refers to this protein that is implicated in neuronal transmission and maturation, specifically in presynaptic vesicle recycling. In humans, there's a lot of point mutations that will give you different outcomes, like hearing loss, epilepsies, or the DORS syndromes that is epilepsy plus mental disabilities. In our case, we will put two flux regions inside this protein, so when we apply tamoxifen to the animal, we are able to create a severe defective protein that is shorter than the original one. And this will allow the accumulation over time of this protein in the brain. This defective protein will lead to the accumulation of endosomes at the synapses, and around 28, 30 days after tamoxifen administration, mice will have a full epileptic phenotype. The good thing about this model is we have a lot of spatial and temporal control about when seizures are occurring, and we can track them very well. Right? So now that we have this, so what is the aim of this project? The aim of this project is trying to directly modify the expression of this gene to affect epilepsy. But first, we need the validation of sleep as a sensitive measure to track epilepsy. So we need to know, having a healthy animal, that when we induce epilepsy and his conditions start progressing towards the epileptic phenotype, we are able to track that with some sleep metrics. For that, I will show you later results about this part, about EEG activity mainly. And then the second phase, that is what we are currently doing, is using nucleic acid therapeutics, specifically antisense oligonucleotides. We are going to try to modulate this gene expression in the long term to help with epilepsy. So yes, again, supporting the long-term recovery by modulating your stress using sleep to track the recovery. And these are the results. So here I'm presenting results of EEG spectra 2 weeks after the tamoxifen administration. We can see how at this stage there's no behavioral manifestation of epilepsy. Both animals are behaviorally indistinguishable from one another. But still, if we analyze the EEG spectra, there's a massive increase on this group. We are analyzing why is the case, but we know it's not an artifact. 1 month later, or 4 weeks, when the epileptic phenotype is present, animals are having seizures, they're having interictal spikes, we see this spectra is renormalized. And when we look at the 24 hours profile of sleep and wake, we can see that at 2 weeks, there are no significant difference between groups. So I must say, this is a pilot study. The size is very small. It's an N of 7 animals. So that will explain why it's a bit, But still, the thing is, 4 weeks after tamoxifen administration, we have a clear delayed onset of sleep in the epileptic group that we can see very clearly. Then, when we analyze the occurrence of interictal spikes, we see that during wake, not even at 2 weeks or 4 weeks, we have seizures, and that is correlated with some types of human epilepsies. And the main difference is that during non-REM, we see that there is a massive increase in the occurrence of interictal spikes. That is also following a sleep-wake pattern, because the animals are in a 12-12 light cycle. So at the end of the light cycle, when the sleep pressure is lowest, we have the highest amount of interictal spikes. During REM, we also see some of this, but we are focusing mostly in the non-REM phase at this point, because even if we see some, they are very rare and it's difficult. So temporal distribution of these interictal spikes will correlate with some types of human epilepsies. And that's why I mentioned before that this has to be a good model, because despite being a nocturnal animal, it's allowing us to see a temporal dynamic of this epilepsy. So what is left is phase 2, is the generation of this treatment. We have successfully developed antisense oligos against ATF4 that I mentioned was the kind of gatekeeper between the restorative function of the unfolded protein response and the apoptotic function of it. And we are completing now the in vitro screening. The candidates that will survive this in vitro screening will be tested in vivo, and those that are the most effective with less toxicity will be the ones we will use with this animal model that we just validated. And this is my group. I'm at Oxford at the group of Professor Vladislav Ryazowski. And my poster is the 111, easy to remember. So if you want to continue the discussion, I will be very happy to keep chatting about this. Thank you very much.
Thank you very much. Do we have any questions?
Yeah, thanks, Chris. It was a lovely talk and a huge amount of information.
Yes, I'm sorry.
Don't apologize. It was great. So my question was, and I hope you didn't say this already, are there any particular cell types in the brain which are more or most susceptible to ER stress? Just for context, I'm thinking because you've got this ASO strategy which doesn't have much cell specificity.
This is currently one of the questions in the field, because right now, for example, the data I presented about the transcriptomic analysis is done taking like the entire tissue, so we take chunks of the cortex. So we are not distinguishing between cell populations, but it's one of the things that is under discussion at the moment. There's not a clear answer, but yeah, it's a very good point, because if we want to really make it work, we need to go more cell-specific.
I don't know if I missed this, but you mentioned that one of your next steps is to screen antisense oligos in an in vitro model.
Yeah.
What in vitro model were you planning to use to do this?
Oh, yes, neuronal cultures. Yes, wild-type mouse neural cultures.
Okay, and what sort of output are you looking for with that?
We are looking for those who are able to be inserted into the neurons are not toxic enough that dysregulate the normal function of the neurons, because the ER stress pathways are very sensitive to a lot of dysregulations. It's one of the main reasons, for example, we don't go with viruses, because that will really trigger the ER stress. And it's a bad idea if already the levels are very high because of epilepsy. So we're trying direct gene manipulation with this.
Okay, so your toxicity screen is in cultures, and then you're going to do your efficacy screen in the in vivo model.
Exactly.
Thank you.
Yeah.
Hi, great talk, thank you. I was just wondering if you'd kind of seen any correlation or expected to see like a link between sort of the severity or the number of seizures, and then the like the extent to which you're seeing this disturbed sleep, and then also if you'd had the chance to look at any of the specific sort of sleep signatures. So you mentioned the slow waves before, or spindles or something like that.
Yes, so we are already detect things. So for example, these mice are very susceptible to sleep deprivation. They start having uncontrollable continuous seizure after 3 hours of sleep deprivation. And if you don't disturb them, then normally the seizures, the full-blown seizures, happening around light onset. It's normally the time of the day that they have it. But it's not a circadian thing, because when we shifted the time, they still kind of have it depending on the amount of sleep they previously have. But it's difficult because sometimes circadian and sleep homeostasis are very intertwined. So it's a very difficult question. But we try our best, and we see a temporal pattern across the 24 hours for seizures and interictal spikes. That normally when you have more interictal spikes, you have less seizures and otherwise. So,
We've got time for one more question. Anyone has one? Okay.
Yeah.
Oh, yeah.
Actually, I do have it. I'm interested, is there any differences between, so a lot of research now is focusing on the differences with males and females within the research. And also, have you looked at the impact of age as well when you're modeling?
We haven't yet. We have the data, but we haven't made any distinctions. What I have shown you is an N of 7 males and females.
Okay.
We haven't made that distinction yet. But yeah, it's something that is in the making, because we need more numbers.
And looked at age as well?
Age as well.
Okay.
But right now we are working always with young adults, because this is a conditional knockout. So we wait 7 weeks and then we induce their epilepsy.
Okay.
So yeah, right now, especially for the treatment, that's important, because we don't know when it's going to be affected. We don't know if we want to apply the treatment before inducing the epilepsy. So because the oligo stays in the brain for around 6 months. So we can play with that. We can say we inject first and then we induce epilepsy, or the animal's already epileptic and then we inject. We don't know what is going to be more effective, but we need to try all possible combinations. Yeah.
Thank you. Yeah, thank you very much.
Okay, next up, I'm going to introduce Kate Hills. So Kate is currently working as a postdoc at the Institute of Neurology, University College London, and she is about to complete her PhD that she conducted at the University of Manchester with Dr. Rob Wykes, studying basically the effect of glioblastoma on epilepsy. And it's this that she is going to be talking about today. Okay. Welcome, Kate.
Yeah, so hi, as Amy said, I'm Kate. So I currently work in the Richardson lab, but all of this work that I'm going to present today was done at the University of Manchester in the Wykes lab and the Centre for Nanotechnology and Medicine. So I'm basically going to give you a bit of a roundup of some of the work that I performed during my PhD, which was focused on exploring hyperexcitable peritumoral networks in a novel model of glioblastoma-related epilepsy. So in terms of where we'll go with this talk, I'm going to give an intro into brain cancer and epilepsy, and then talk about kind of what we're doing to research glioblastoma-related epilepsy. I'll talk about what we found, and also the implications that this has for epilepsy generally and in brain cancers. So glioblastoma, or GBM, is a WHO grade 4 glioma that carries a notoriously poor prognosis. So median survival in patients is typically between 12 to 15 months with current standard of care, which has really remained unchanged for over a decade now. Seizures are a common comorbidity in this disease, with up to 60% of patients experiencing tumor-associated epilepsy at some point in their disease course. So this can happen when the bulk tumor is still in situ, post-resection when there is tumor recurrence, and in the end stage of the disease. And it's generally this region of brain, like healthy brain, around the tumor, called the peritumoral border, where the epilepsy is thought to originate from. We know from clinical literature that tumors in the frontal and temporal lobes, and especially those with a cortical localization, carry the highest risk of seizures. Also recently, preclinical work and some clinical studies have shown that spreading depolarizations, which are another network phenomena, have been shown to occur, which this phenomena has been associated with the pathology in a lot of other neurological diseases, such as stroke and/or familial hemiplegic migraine. So how are we looking to study this? So what we've been doing is establishing an immunocompetent model of glioblastoma-related epilepsy. We chose to go with a transplantation model, which is basically where you inject GL261 cells, which are a mouse glioblastoma line, into a region of the brain, and we picked the entorhinal cortex. We chose this model because it offers precise control of tumor growth and also high reproducibility, and it's very standardized in the cancer space. So, like I said, we chose the posterior entorhinal cortex, and this was mainly because of its location and the reciprocal connections with the hippocampus and cortex, so that it wouldn't ablate serious regions of the brain before the epilepsy had chance to form. How we were characterizing this model is using different imaging modalities like bioluminescence imaging and MRI, and doing post-hoc immunostaining, and also concurrently doing electrophysiology, both chronic and acute experimental paradigms. And this was basically to evaluate seizure and spreading depolarization presence with tumor growth. So what we were able to kind of glean from our imaging studies, specifically in MRI, was that there's a distinct pattern of growth and cortical infiltration. So what you can see here is an anterior sort of slice, coronal slice in the mouse brain, and a posterior slice where we injected. So at day 7, this is the injection tract, and this is the region of the tumor implantation. At day 14, you can see it's begun to grow in this same region, kind of up the injection tract. And by day 21, it's encompassing this entire region and beginning to extend anteriorly with some added mass effect and some infiltration, well, definitely infiltration, of the visual cortex. Even though it's not particularly clear in this image as well, you can see that there are some regions of like edema and necrosis and hemorrhage. And when we quantified what this looks like in terms of using tumor volume as a measure of growth rate, we found that there was a significant increase in the tumor volume in the later stages of the model. And we corroborated this using bioluminescence imaging, which we found the same data. We also took these brains and applied hematoxylin and eosin staining, which is basically like this, so the tumor is in the deep purple. And what we saw was progressive expansion of tumor into gray matter with some infiltration of white matter, kind of what you can see here. This also corroborated our MRI data showing areas of edema and disorganized vascularization. And we also saw significant disruption of subcortical structures such as the hippocampus, which is important for when we're thinking about generating an epilepsy model. But we needed to look at the seizures, not just the tumor, so we employed 24/7 single-channel video ECOG to confirm seizure presence. And basically what you can see here is a reference electrode and a single point of recording in the secondary visual cortex, which is, we chose this implantation site based off our previous MRI studies, kind of trying to estimate where the peritumoral border would be in the later stages of the model. And what we observed was multiple types of epileptiform activity. So we saw multiple different kinds of seizures that were evolving and happened over tens of seconds. And we also saw intermittent periods of interictal activity. Interestingly, this interictal activity increased over the course of the model. And what this maybe tells us is that the tumor and the peritumoral border is growing into our electrode recording site because we only have a single point of recording. We also found that seizures appeared to cluster in this model, and they were more likely to occur within 3 hours of each other. And when we looked at the power of these seizures, we found that the last seizure in a cluster exhibited significantly higher total power. This is interesting not only for glioblastoma-related epilepsy, but also for epilepsy in general, because clustered seizures happen a lot in different syndromes, and how the seizure evolves over the cluster may have implications in terms of how they're treated. So any information about this is pretty useful. So to sum this slide, we found that GRE mice display spontaneous epileptiform activity and clustering of seizures, but really how does epileptiform activity change with tumor progression and location? To look at this, we employed multi-electrode head-mounted transmitter devices to record from the peritumoral border and healthy brain. So we implanted a contralateral electrode, which is the one in purple there, and 3 electrodes on the same hemisphere as the tumor, with the green and the blue being in peritumoral border and the somatosensory cortex being in what would be healthy brain to begin with, but clearly isn't there. And what we found was that there were intermittent periods of interictal spiking that were initially restricted to peritumoral electrodes. So we didn't see any in the somatosensory cortex, and there was no generalization to contralateral. As the model progressed, we saw that there was an attenuation of the signal in the electrode closest to the tumor, and that these spikes became generalized, which is, you can see, kind of blow up here. and involved the contralateral somatosensory cortex. This has also been seen in one other genetic model of glioblastoma-related epilepsy, so that's quite exciting. But what we managed to do is show that progressive expansion of the peritumoral border is observable longitudinally, which is not something that's been really looked at in detail like this before. And it's also, we've been able to show that this can be indicated by interictal spiking, which is particularly useful clinically because this kind of, because during resection, when they take out the bulk tumor, oftentimes ECOG strips will be applied to kind of identify tumor-infiltrated regions, and it's been shown that areas identified as having interictal spiking, once they've been resected, there are better recurrence outcomes. We also had video, so we used telemetry sync video to characterize the distribution of behaviors during seizure. So how these evolved was typically they began with staring or behavioral arrest. This progressed into things like neck jerks, which then moved forward into clonus, like forelimb clonus, hind limb clonus, and dystonic posturing. Towards the end of the seizure, we saw myoclonic jerks. And interestingly, later on in the model, we found that there was sometimes prolonged periods of postictal depression. Generally, we found that freezing and behavioral arrest usually signaled the initiation of a seizure, but occasionally occurred alone. And generalized seizures were, well, focal to bilateral generalized seizures were dominated by neck jerking, limb clonus, and a loss of postural tone. This kind of phenotype is consistent with other rodent models of epilepsy and also with the clinical presentation in glioblastoma-related epilepsy, which is dominated by focal to bilateral seizures. Like I just briefly talked about postictal depression, this is interesting for us because postictal depression has often been associated with increased seizure severity. Postictal depression generally is a transient suppression of cortical activity following a seizure, and so we wanted to look at this more in our model. So what I did is I developed a semi-automated pipeline that basically set a criteria for the postictal depression, which was a period of longer than 8 seconds below 50% baseline total power. We found that 33% of our seizures were associated with a postictal depression, and that these seizures generally had a higher power than those without. We didn't see any association with seizure duration, but when we looked at clustered seizures in that group that we previously analyzed, we found that the last seizure in a cluster had a higher frequency or was more likely to be associated with postictal depression. So this maybe kind of tells us that postictal depression, in at least our model, appears to be linked to seizure intensity, not necessarily duration. But this also asked a wider question of, because we're recording above 0.3 hertz, which we're recording with a high-pass filter, is this silencing of the EEG trace indicative of other phenomena, potentially like spreading depolarization? So spreading depolarizations are a slowly propagating wave of neuronal and glial depolarization, and they are associated with a large disruption of ion homeostasis and can be followed by a suppression of electrical activity. To look at these, we couldn't use the old transmitters we were using, and we had to use DC-coupled longitudinal telemetry devices, which are these HMTs that I talked about just before. These can record seizures and spreading depolarizations in our model concurrently. So when we did this, we observed spontaneous spreading depolarizations, which were often detected first at the peritumoral electrodes, which are these ones here. And we also observed seizure-associated spreading depolarizations with the progression of the model. So this is kind of what you can see here, leading into the spreading depolarization. This didn't always require seizures to be generalized either to induce the spreading depolarization. We did some more in-depth analysis on the behavior that was present during spreading depolarizations, but in the interest of time, I won't go through it, so I'll just go over the main points. But they were generally behaviorally dominated by behavioral arrest and dystonic posturing, which this hasn't really been previously reported, but is interesting in the context of our model because our tumor is kind of close to the cerebellum. So the only other, I think there's 2 other papers in the literature that discuss dystonic posturing in the context of spreading depolarization, and this is when they found the spreading depolarization propagated into the cerebellum. We also occasionally saw myoclonic jerks toward either in the middle or towards the end of the spreading depolarization. So we validated previous work and clinical studies that in our model, spreading depolarizations occur spontaneously in glioblastoma-related epilepsy. But what we really want to know is how do spreading depolarization properties change relative to distance from tumor? And to do this, we employed graphene solution-gated microtransistor arrays. So graphene solution-gated field effect transistors are microtransistor arrays. They can record in a wide bandwidth, covering infraslow activity and high-frequency activity concurrently. They're different than the electrodes I was using before in that they are active. So changes in extracellular voltage in the neural network modulate a current that flows through the transistors. They come in square shape, rectangular shape, and also some intracortical configurations. But the ones I was using had 16 recording channels, and they're all flexible and biocompatible, which is great in a brain that is distorted by a glioblastoma. So we wanted to look at whether, now we know that they occur spontaneously, whether evoking these spreading depolarizations could be kind of a functional readout of tumor-associated hyperexcitability. So we utilized a pharmacological induction of spreading depolarization via superficial cortical injection of potassium chloride. This is a very standard method of inducing spreading depolarization, both in vitro and in vivo. So how we did this experiment was I implanted the glioblastoma at day 0, and then after much optimization, we settled on the days between 16 and 18 within the model, a point at which the network was hyperexcitable and we had previously recorded seizures at, but where there was no mass effect from the tumor. And what we found in this is that when we injected identical stimuli, so 1 microliter, 1 molar KCl, into glioblastoma mice, this evoked repeated spreading depolarizations, versus the same stimuli only evoked one repeatedly in sham mice. This is interesting not only in this model, but also because other models of other models like familial hemiplegic migraine mice, which are known to have SD susceptibility, display the same thing. This, however, showed us potentially that GRE mice do show an increased susceptibility for spreading depolarization, but it also begged the question that are these recurrent SDs due to potentially impaired potassium clearance? Are they due to increased cortical sensitivity to injury because we have to inject the potassium chloride? Or potentially is it the same spreading depolarization circling back around the hemisphere? So we employed a different model to induce spreading depolarization that we were hypothesizing would reliably induce a single spreading depolarization so that we could kind of tease this apart. And this is using optogenetic induction, which has been standardized in across the literature for a while now. So what I would do was I would inject channel rhodopsin virus about 3 to 4 weeks before I implanted the glioblastoma, and then at the same recording point, I would then induce spreading depolarizations in a stepwise paradigm. So I would increase the LED power and increase the duration of the stimulation to try and reliably try and test the threshold for spreading depolarization. And I was classing a threshold as 3 reliably evoked spreading depolarizations. This didn't appear to change the way the tumor grew. The red is the channel rhodopsin, and the dark blue kind of here in DAPI is the tumor. And what we found is that glioblastoma-related epilepsy mice generally required a lower LED power and duration than sham mice to induce a spreading depolarization. They also demonstrated higher propagation velocity, which is kind of this dot plot here, where you can see this is distance from tumor, and you can see that there is a higher propagation velocity here, and there was a lower amplitude of the spreading depolarization in peritumoral areas, which are both quite interesting findings. This is consistent with literature in other models where potassium clearance is impaired, showing that you have a higher SD velocity. So we showed that spreading depolarizations display spatially dependent changes in their characteristics in glioblastoma-related epilepsy mice, and we know that this is probably related to potassium, but one of the big hypotheses in glioblastoma is glutamate, and could this potentially be related to glutamatergic action? So we're nearly done, just a couple more slides. You're nearly through. So the glutamatergic hypothesis in glioblastoma-related epilepsy centers around the idea that tumor-mediated disruption to glutamatergic neurotransmission acts both in favor of glioblastoma growth and epileptogenesis. Recent work, seminal work by the Monge group in the US and the Winkler lab in Germany have shown that glutamatergic neuronal glioma synapses exist within the tumor, and if you express channel rhodopsin in either glioma cells or neurons and excite them, these activity at those synapses can drive invasion and progression. So it's clearly a key component. What we chose to focus on is the astrocytic glutamate reuptake transporter, GLT1. It's well established that the glutamate antiporter, system XC, is overexpressed in glioma cells and absolutely pumps out glutamate into the synaptic cleft. So why isn't this, there's clearly something wrong with this, and why isn't it working in glioblastoma? So what's generally been found in GBM cell lines and tissues is that there is a reduced GLT1 expression, and specifically tumor and peritumoral tissue from GBM patients with seizures shows a more significant reduction in this transporter than those without seizures. So clearly it's involved somewhere. And the result of it being involved is that glutamatergic signaling at neuronal and gliomal NMDA receptors and AMPA receptors is potentiated, which can have knock-on effects on seizure development and spreading the polarizations. When we looked at GLT expression in our mice, we found that GLT was significantly reduced in tumor regions, which the GLT is in pink, and the DAPI is indicative of the tumor because it has hypercellularity, and GFAP staining is in green for reactive astrocytes. When we looked closer at this, you can see that in the contralateral hemisphere, there are these pink dots surrounding the astrocytic processes, but in the peritumoral border, this is almost completely absent, with the astrocytes displaying a more reactive morphology. So we found, in addition to the reduction in GLT, that GFAP expression is increased in peritumoral regions, and the same with the GLT. So clearly reactive astrocytes are present in the peritumoral border, but they do demonstrate reduced GLT expression. So something is going wrong here, and it is indicative of disruptive glutamate clearance, which definitely will contribute to the tumor progression, seizure presence, and spreading the polarization in this model. To sum up and look at implications for epilepsy and brain cancer, spontaneous seizures in our model appear to be driven by increased cortical infiltration and are associated with dysfunctional glutamatergic neurotransmission. We saw that GBM-induced ion gradient disruption increases peritumoral susceptibility to hyperexcitability and spreading depolarizations, as shown spontaneously and through pharmacological and optogenetic studies. In the context of cancer neuroscience, this work only really enforces the idea that investigating neuron-cancer interactions is completely crucial, because neuronal activity profoundly influences tumor initiation, growth, and progression. The tumors don't just develop in isolation. And in terms of the future, where this field should be going, and as more neuroscientists get involved in cancer, we should be looking towards whether targeting the tumor-adjacent microenvironment can provide an alternative therapeutic approach to influence tumor growth and susceptibility to these comorbidities. And could this help address the significant barrier to treatment efficacy that's currently posed by the heterogeneity of glioblastoma? Ultimately, investigating this interaction between seizures and spreading depolarizations in preclinical glioblastoma models will show us whether targeting network hyperexcitability will delay tumor progression and have favorable outcome on GBM prognosis. And I'd just like to say thank you to everyone that's contributed to this project in the Wikes Lab, the Center for Nanotechnology and Medicine, our collaborators that helped produce and with the analysis of the GFETs, Open Source Instruments, who is the longitudinal telemetry, the staff and the technicians, and of course the funders. Thank you very much.
Thank you. Great talk.
Thank you.
I had a few questions, if that's all right. First of all, did all of your mice that you injected end up having epilepsy or seizures?
So not all of them developed seizures. We had a really good response rate of about 90%, which was probably down to missed targeting of injection,
So
the way the tumor
were
grew.
there any certain tumor volumes or infiltration to any anatomical parts of the brain that you noticed kind of induces seizures more than those that didn't?
So it's because we weren't doing MRI and electrophysiology in the same cohorts because the electrodes aren't compatible. We couldn't know whether the mice that were undergoing MRI had the same seizure progression pattern as with the electrophysiology. We assume they do, and it's reasonable to. But ultimately, what we looked at from the MRI and basing it on the time points at which they were having seizures in the longitudinal cohort, we saw that kind of in the middle to late stage of the model, they began to have seizures, but especially when we started using the multi-electrode array, head-mounted transmitters, we found that the interictal activity was there for quite a while before, and the spreading depolarizations occurred before any seizures even happened. So in terms of like anatomical structures, it wasn't really, it was more the infiltration of the visual cortex as opposed to any kind of distortion or mass effect, which was observed later on.
Okay, and last question. You alluded to this on your previous slide about neuron, cortical, and GBM interactions.
Mm-hmm.
There's a really good paper from North America which showed that the GBM cells kind of hijack the networks, and they send these pulses towards the GBM for it to grow.
Mm-hmm.
And one of the biomarkers is spreading depolarizations. So my question is, did you see any correlation between SDs and tumor aggressiveness or any synapse kind of biomarkers that you might have looked for in antibodies? Or
So we didn't do any synapse staining, but it's definitely like that would be really interesting to do, I think, to look at whether that's changing in mice that we have definitely observed spreading depolarizations in. We didn't observe any particular difference in terms of the tumor aggressiveness, but that's definitely something that would need to happen more. It would be really interesting to try and compare to a cancer model that doesn't, to compare to a different injection site that doesn't have seizures or spreading depolarizations with the same number of cells, and see whether having spreading depolarizations at the start of the model or whether you induce them before seizures have happened, whether that then influences survival rate and things like that. But we personally didn't do any of that work, but it is really interesting. Thank you.
We should declare that we're neurosurgeons. That's why we're asking these questions.
Cool.
So the data that you showed about the inextricable link between the seizure formation and the tumor growth, if you look at epidemiological data, there's no difference in glioblastoma rates between people with idiopathic epilepsy and not, and there's no difference in response rate to treatments for people who are on anti-seizure medication versus not. So how do you reconcile that difference between your animal model and the real life data?
So you mean in terms of the fact that people with epilepsy aren't more predisposed to?
Yeah, there's no difference in the rate of tumor development if you've got epilepsy for one reason versus the rest of the population.
Yeah, so it's a good question. So in terms of people with epilepsy for different reasons not being more predisposed to having glioblastoma, a lot of the mechanisms behind the epileptogenesis are the same, but there are a lot of things that are different in glioblastoma, and a lot of things that I believe, and a lot of other people within the literature specifically, I don't know if you've read any of like Nobel's papers, that talk about in the early stages of the glioblastoma development, how this is impacting the network connectivity and things like that, to then go on and develop the epilepsy. So I don't think it's necessarily so based on this excitation, inhibition, the classical way of thinking about it in epilepsy. I think there are a lot of things that are specifically glioblastoma driven that contribute to that. But it is really difficult to tease apart because it occurs at different points in the tumor course, and there are most likely different mechanisms underlying each point that occurs. So like in the end stage of the model, you're probably going to get tumors from the mass effect. It's not going to have been the epileptogenesis that's growing over time. But it is something within the literature that a lot of people like kind of go back and forth about. It is really interesting. So thank you.
Oh, thank you.
Do we have any more questions? I have a question for you. So you said that more severe seizures are associated with, or more likely to be associated with, spreading depression. or spreading depolarizations. Do you have any theories as to the mechanisms of why a more severe seizure would be associated with a spreading depression? Sorry, that's quite a mean question.
So on the face of it, I think, because we showed that kind of, well, post-ictal depression, for one, doesn't automatically infer spreading depolarization, then a lot of papers have found that that's not necessarily the case. But in terms of us showing that the intensity of the seizure is maybe what drives the post-ictal depression, and whether this then translates to spreading depolarizations, yeah, on the face of it, it would probably be the fact that if you have a more intense seizure, you're dumping out loads of potassium, you're dumping out loads of glutamate, cells are going off left, right, and center, and potassium is known to be a big driver of spreading depolarization. And when you're in sort of a disease state like glioblastoma, where the surrounding tissue is already severely metabolically compromised, and you're not able to take up any of those ions or any of those molecules, you're basically just inviting a spreading depolarization to happen. And then any sort of knock-on effect kind of with the expression of NMDA receptors, the expression of AMPA receptors, but most likely NMDA, will then influence the propagation and how quickly you recover from spreading depolarization and things like that. So I think it's a big party of dumping out loads of stuff to then influence the next lot of neurons that in a brain that can't recover, you're just going to go and go and go and go, if that works.
Okay. Do we have any other questions? Okay, then thank you very much.
Thank you.
So I'm going to introduce now Dr. Nicolas. Dr. Nicolas Mariscal is a research associate in the laboratory of Professor Benedict Berninger at the Center of Developmental Neurobiology, King's College London. He joined Dr. Berninger's laboratory after successfully competing for a prestigious postdoctoral fellowship of the Human Frontier Science Program to study in vivo reprogramming of glial cells into neurons, an innovative approach for neuron regeneration. Recently, Nicolas was awarded the Emerging Leader Fellowship from the Epilepsy Research Institute, UK, to lead his research plan to advance this technology in the context of temporal lobe epilepsy.
Okay, good afternoon, everyone. Thanks, Prius, for the introduction. I would like to thank the Epilepsy Research Institute, UK, for giving me the opportunity to present my work here today, the work that I have been doing actually here in the fourth floor at the Center for Developmental Neurobiology at King's. And the problem that I'm focusing on is mesial temporal lobe epilepsy, which is one of the prevalent forms of focal epilepsies. MTLE is initiated by a precipitating factor, and then it's characterized by the occurrence of hippocampal recurrent seizures that can be measured by EEG recordings only in the hippocampus and not, for example, in the cortex. Unfortunately, you also hear from Annie's talk that many patients are resistant to anti-seizure medication, and resection of the hippocampus may be for them the only option. So this really illustrates the urgent need for novel therapeutic strategies to treat MTLE. We have a close look of the histopathology, what's going on in the hippocampus. Well, many patients have what is called hippocampal sclerosis, which basically have massive neuronal loss in CA1 and CA3, as you can see here, healthy hippocampus comparing with the MTLE hippocampus from MTLE patients, so massive neuronal loss in CA1 and CA3, and also dispersion and hypertrophy of granule cells. But important features for my talk today are also the presence of massive reactive gliosis and also the death of inhibitory GABAergic inhibitory interneurons. You can see here staining for interneurons in healthy tissue, in healthy patients. You can see spread there, but in epileptic sclerotic hippocampus, only some process survive. And this makes an excitatory inhibitory imbalance that is one of the reasons behind the focal seizures. So this gives us the idea that maybe restoring inhibition but generating new inhibitory interneurons in the hippocampal sclerosis can be a novel strategy for seizure control. So how can we do this? Well, in the lab, we are working a novel strategy for neuronal regeneration, which is called linear reprogramming. So basically, the idea is simple. We are trying to convert the endogenous reactive glia into GABAergic inhibitory interneurons. And the strategy that we use to do so is by the overexpression of key regulators of neuronal fate. In this case, key transcription factors that play important role during the differentiation of GABAergic interneurons. So which transcription factors? Well, you're going to hear a lot in my talk of our favorite transcription factor, which is called ASCL1. And basically, ASCL1 is highly expressed during embryonic development in the ventral telencephalon, as you can see here, in the region in which GABAergic inhibitory interneurons are born and are generated. And it has been shown in previous literature that plays a really important role during the differentiation. So we asked whether, okay, can we make reactive glia become interneurons? So the first question was if we can do this in the context of MTLE. And for this, we use a well-established preclinical mouse model of mesial temporal lobe epilepsy, which consists in the injection of intrahippocampal injection of kainic acid, and here in adult mice. And here I need to mention our fantastic collaborator, Dr. Christoph Henrich and his group in Lyon, who really pioneered the use of this mouse model in the context of to study glia to neuron conversion. So basically, the mouse model recapitulates many features of MTLE with hippocampal sclerosis, as you can see here. So in the ipsilateral hemisphere, there's massive loss in CA1, massive neuronal loss in CA1 and CA3, and also clear granule cell dispersion and hypertrophy. And importantly, there is massive death of inhibitory interneurons, which are labeled here with the GAD67 GFP mice in white, and as you can see, they are absent in the injected hippocampus. So then our strategy to reprogramming was after the induction of epilepsy, injection of retroviruses encoding the overexpression of transcription factor to target reactive glia. So retrovirus control, empty vectors without the transcription factors, and retrovirus expressing ASCL1, and also in this study we use DLX2, which is another really important transcription factor for the differentiation of GABAergic interneurons, and perform the analysis later. And what actually Christoph found that, yes, when we use control retroviruses, you can see the cells retain glial morphology and they do not express neuronal markers. However, when we target cells and we overexpress ASCL1 and DLX2, as you can see here, the cells acquire neuronal morphology, bipolar neuronal morphology, and really importantly, around 60 to 80% become GABAergic interneurons, as are labeled here again using the GAD67 GFP mouse line. So this was really exciting for us, but then the next question was, are these induced interneurons functional? Can they make functional synapses into epileptic postsynaptic neurons? And for this, what I did was applying an optogenetic approach to answer this question. So basically now, after the induction, so here is the experimental design. After epileptic induction, we inject and to overexpress our transcription factor, but also we inject a second retrovirus expressing channel rhodopsin. So we express channel rhodopsin in our induced neurons. And then I performed patch clamp recordings in hippocampal slices. In one set of experiments, only patch the induced neurons to see and to study their functional properties, to see whether they can fire action potentials. And in a second set of experiments, patch only granule cells, which are the postsynaptic cells, and activate by blue light our induced neurons to see whether we found responses on the postsynaptic granule cells. And what we found was that when we patch our induced neurons, as you can see here, indeed, they are functional. They can fire action potential repetitively. They show some maturation. And also, we tested that we can indeed activate it by light. When we shout light, we can depolarize and activate the induced neurons. And then, what we found patching the postsynaptic granule cells, we activate the process of all our induced neurons surrounding a specific granular cells that we patch, and what we found that indeed, as you can see here, we can found inhibitory postsynaptic potentials in this case, also inhibitory responses in the granular cells that were blocked specifically by GABAcin, which is a specific blocker of GABA-A synaptic receptors. So this was the first demonstration that neurons induced from glia are functional and can make functional synapses into postsynaptic cells. That was really, again, exciting for us, but the last question was, okay, can our induced neuron have an impact on the seizure control? And for this, what Christoph did now was the following. So the experimental design is here after the induction of epilepsy and after the injection of our transcription factors. He performed EEG recordings in the mice. And what we found was that, as you can see, comparing control animals which were injected with empty viruses, the seizures are here everywhere, while in the treated mice injected with ASL1 and DLX2, the seizures were reduced, significantly reduced. And here is the quantification. So in red are the treated mice. As you can see, the number of seizures per hour and the time in seizure that the animal spent were significantly reduced. Again, this is published, so you can read more about this work. And this is fantastic for us, but we still think that there is room for improvement because, as you can see here, animals are still, and the mouse still have seizures. They are not seizure-free, and we really wanted to reach this point. So, and why can we do this? So we think about many possibilities, and one can think the subtype of interneurons that we are generating with our reprogramming strategy. So if you're not familiar with interneurons, they come in different flavors. So we test some antibodies to see whether we found some subtype specification with ASL1 and DLX2, and we found that indeed some cells express, for example, BAP or somatostatin or MPY, as here is quantification. But we were really intrigued about the absence of induced neurons with hallmark of fast spiking parvalbumin interneurons. So if you are not familiar with these interneurons, they are the most abundant subtype of interneurons in the hippocampus and also in the cortex, and they play a really important role in maintain, and they exert a really powerful inhibition in postsynaptic cells, and they are really, really vulnerable to the injection of kainic acid. As you can see here, well, you can see, I hope you can see in many cells of PV, the normal, how they really make enormous amount of synapses in the granular cell layer, but they are really vulnerable in the ipsilateral side. And also a key feature, a key hallmark of this subtype of interneurons, they can fire, this is what they are called, fast spiking, they can fire action potential in really high frequency, above 150 hertz. So now the hypothesis that we have is that maybe if the induction of fast spiking parvalbumin interneurons will improve seizure control in the mouse model of MTLE. But the next question was, okay, how can we make now specifically fast spiking parvalbumin interneurons? So in the lab, we have been working in another experimental model to study glia to neuron conversion, which is a targeting of proliferative glia in the P5 mouse cerebral cortex. And this is why the reason of this is because there is a massive, at this time point, there is a massive gliogenesis during the first week of postnatal, in the first postnatal week in the cortex. And here, what we did was actually to tune a little bit ASL1, because ASL1 activity is somehow regulated by post-translational modifications, including, for example, phosphorylation. So the preventive phosphorylation of ASL1 has been shown that promotes GABAergic neurogenesis in progenitors during embryonic development in the cerebral cortex. So we prevent the phosphorylation using a phosphomutant side version of ASL1, which we call ASL1 SA6, which basically have 6 serine motifs mutated by alanine, and this prevents the phosphorylation. So we asked the question, what happened when we use this phosphomutant side version of ASL1 comparing in the cortex comparing with ASL1 activity? And what we found, interestingly, was that some of our induced neurons using the mutant version of ACL1, ACL1, express parvalbumin levels, as you can see here, a high proportion, around 20-25%. And we also found these cells in layer 1 of the cortex, which you will not find parvalbumin positive interneurons in this layer, confirming the authenticity of our reprogramming strategy. And remarkably, most of or a high proportion of the cells that we managed to patch and to study fire action potentials in a really, really high frequency above 120 Hertz, which is a hallmark of fast spiking interneurons. So again, this is also recently published, so you can read more, and I'm happy also to then chat about this work. So now we believe that we have a new tool to generate these interneurons in the context of MTLE. So I'm really grateful to Epilepsy Research Institute UK, because in the next years I'm going to be working in this idea, basically test this hypothesis. We know that AC1 and DLX2 can make interneurons that express somehow somatostatin, MPY, and BAP. But the question is now with our ACL1, ACL6, if we can make fast spiking parvalbumin interneurons. So we're going to pursue three aims. So the first one is, of course, the subtype identity and the characterization of the neurons, of the induced interneurons that we can get with our ACL1, ACL6. The second will be assess the synapses, how robust and how, and if they can also inhibit postsynaptic granule cells, epileptic cells, and of course, try to analyze the impact on seizure control. So we started with this a couple of months ago, but so today in the last part of the talk, I'm going to present some preliminary results about the subtype identity. And the first basic question actually was, can we indeed make induced interneurons using our ACL1, ACL6? Because we also know that reprogramming is quite cell and context dependent. So different transcription or the same transcription factor can have different outcome depending on the cell type that you target and the environment. So we made our first injection. So after again, here is the experimental design. After the induction of epilepsy, we inject our transcription factor, our viruses overexpressing now our ACL1, ACL6, and analyze tissue in short time points, and now only 2 weeks later to see what's going on. And what we found here, that as you can see here in the low magnification, we have many cells target in the hilus and in the granular cell layer. Also, we have cells that can be converted, reprogrammed in near to CA1. And if we have a close look, as you can see here, many cells, or most of the cells, acquire clear neuronal morphology, extend processes that goes through to the granular cell layer, and as you can see, many of them express neuronal markers like NUN. We also test for another neuronal markers like microtubule-associated protein 2, and as you can see here, most of the cells express not only in the cytoplasm, but also in the processes in the dendrites, suggesting, so this really, our preliminary results suggest that ACL1, ACL6, as it is in the cortex, looks to be really powerful to make neurons from glial cells. But another interesting question for us is the source of our induced GABAergic interneurons. And because I mentioned that we wanted to convert reactive glia into neurons, and maybe you all think when I say reactive glia, you think about astrocytes. But there are other subtypes of glial cells in the brain, specifically, for example, oligodendrocyte precursor cells, and they also become reactive after in the MTLE model. So we asked the question, can we make induced interneurons from oligodendrocyte precursor cells? And for this, here is experimental design. We are doing genetic phase mapping. So basically, what we use now is a mouse line in which we can label after tamoxifen injection in green NG2 cells or oligodendrocyte precursor cells. So we inject tamoxifen to label all the oligodendrocyte precursor cells or many oligodendrocyte precursor cells in green. Then we induce the epilepsy, and then we inject our viruses that express ACL1, SA6, and the red fluorescent protein. And we analyzed to see whether we found cells target that become neurons that were also green, suggesting the origin of OPC cells. And what we found, as you can see here, that indeed we found subcells that acquire neuronal morphology, they were phase mapped, and they express neuronal markers. So this confirmed that we can also make induced interneurons from oligodendrocyte precursor cells in the context of MTLE. But one important question for us is, can we make somehow or have some PV signal in some of our induced neurons? So we perform, again, staining, preliminary results, or preliminary staining, and as you can see here, while some cells we couldn't find in some induced neurons PV expression, in other induced neurons we found some PV staining positive cells, and interestingly, we found the staining or the expression of PV in cells that were phase mapped from oligodendrocyte precursor cells. And this is quite new for us because in the previous study that I mentioned, we only made PV cells from astrocyte in the cortex. So this is really interesting for us. As I told you, the source, it can be really important because it will be really great to not only have astrocyte as a possible source to make new neurons, and we now know and we confirm that we can also use OPCs as a source for our new interneurons. Finally, we are also testing the functionality in acute hippocampal slices. So after the injection of after the induction again of the epilepsy, the injection of our reprogramming factors, we perform some patch clamp recordings on the cells, and what we confirm that indeed, and here's one example, you can see the cells reprogramming with ACL1, SA6 in the context of MTLE, can fire action potentials repetitively, and interestingly, they are really well connected. They receive, as you can also see here in the current clamp recordings, but also in voltage clamp, they receive substantial excitatory postsynaptic currents, suggesting that they are really integrated in the circuit. Hopefully, maybe in the next VNA meeting, I can tell you more about their connectivity and also their impact on seizure control. So conclusions or take-home message. Yeah, I show you today that ACL1 and DIREX2 can convert reactive glia into interneurons in the epileptic hippocampus, that these interneurons make functional synapses and reduce epileptic seizures. Also that the phosphoside deficient ACL1 can convert cortical astrocyte into neurons with hallmark of spiking parvalbumin interneurons, and that we also know that the same ACL1 SA6 can reprogram reactive glia, including OPCs, in the context of MTLE into functional induced neurons. I hope also that I convince you that glia to neuron conversion is a valuable strategy to restore inhibition in this context and hopefully to have a great impact on seizure control. Well, just the last slide, one of the most important. I would like to thank particularly Benedict here in the picture, the entire Benedict's Berninger lab, particularly Sophie and Anna, who worked together with me in the story in the cortex, and my recent student, Firzana, who is working in the vulnerabilities, helping me in my project and study the vulnerability of PV in the MTLE mouse model, and of course, Epilepsy Research Institute and the rest of the funding, and thank you for your attention.
So now time for questions. Yeah.
So it feels like quite a simplistic question, but is there some kind of risk that you don't have enough astrocytes left after you've converted them to interneurons? Is there any sort of danger there that you could perceive?
Yeah, so no, it's a great question, actually. So, of course, astrocyte plays an important role in homeostasis, and we don't want to deplete the hippocampus from astrocyte. The number of cells that we target now is comparing with the total amount of reactive gliosis is quite low. So I think it will be unlikely that we target and we convert all. But yeah, it's something that we need to consider. And maybe if this is the case, if we manage to have a huge amount of astrocyte converted, maybe we can combine our strategy with something that makes cells proliferate and make astrocytes proliferate more to maintain the levels. And we also need to think the same about OPCs. But believe me, the number of cells, the number of reactive glia that is there is massive. So it's quite unlikely that. But yeah, we need to consider do not deplete the region of glial cells.
Thank you very much. It's really cool to see the development of the PV neurons. Do you know whether they synapse around the perisomatic area of the excitatory cells? Or
That's a great question. No, we don't know. Yeah, so in the MTLE, we still are in the preliminary phases, let's say. This is, actually, we submitted a grant, but it was rejected, trying to do electron microscopy. That will be one way to know. Because, they should, if we really are making real PV, they should synapse onto the soma. We don't know, but we are planning to study. Yeah, it will be super great to know. Yeah.
Thank you. Fascinating presentation. Maybe I missed something at the very beginning, if you can help me. When you inject your retroviruses with your reprogramming factors, is there any cell specificity of their action? Because you immediately said, I'm reprogramming astrocytes. Great. Why? But then you said you can also reprogram the oligodendrocyte precursors. Is there a chance these factors can go into neurons or into any other cell for that matter, or maybe damaged interneurons and rescue them to be better interneurons? So how do you target this to cell types?
Great question. So I didn't mention in detail, but the answer is about how retrovirus works. So retroviruses can only transduce proliferative cells. So this, so have pros and cons. But in our context, this allows us to only target reactive glia. So interneurons are postmitotic, so we only target proliferative cells, which in this context, and also, as I told you, in our mouse model of postnatal mouse model in the cortex, there are glial cells that are increasing in this context of the cortex. They are increasing the numbers to populate the entire cortex. In the context of MTLE, they are reactive. But they are so we are only targeting cells that are undergoing division when we inject, which this is like a control for us that we only target. Then we cannot so we target everything that is dividing, so astrocyte and OPC, both. That's why we are using the fate mapping to understand which is the source or which different types we can.
Thank you very much. That confirms my previous question to the previous speaker, where I immediately said I know nothing about viruses.
Two probably naive questions. One is the human hippocampus is a much bigger space than the mouse hippocampus. So is there a strategy for getting the virus in there reliably? And the other is that temporal lobe epilepsy is a progressive disease with progressive loss of interneurons. So is this just a temporary fix where the new interneurons are going to get lost as well?
Yeah. So of course, these are two translational questions. Let's say that. So we are in the proof of concept, let's say, phase yet. Yeah, so the use of retroviruses, again, the problem is the number of cells also that we can target, because we only can target proliferative cells, and also the amount they don't express like AAV viruses. Of course, in the lab, we have been trying to use AAV viruses, but we have some issues because and also literature in the field have the same issues of us. We have leakness of this, and we target neurons with viruses that even we're using specific promoters or everything, they end up being leaking. So we are still working with our retroviruses to really confirm and to have more proof of concepts that the system work, and try to refine the use of AV to see whether we can have something that in theory can be then more translational. So the answer is, yeah, we will need maybe another tool, or to think in the size of the human brain. And the second question is, so we know that also it will be great to inject later. And our cells, so far that we know, we have data is already published that our reprogramming cells can be after 7 months there. So which means that even though the animals have seizures, they seem to survive. So yeah, hopefully they can survive. Yeah, well, and it will be great also to try to do our injections later, or maybe 2 or 3 times to replace and have more in the case they are dying. So these are things that we can try. And in our model, as soon as we have proliferative glial cells, we can target and try to make several injections, for example.
Okay, I think we're nearing the end of our session today, and I'd just like to thank Nicholas again very much for an amazing talk, and also we can give every one of our speakers today a really massive round of applause in terms of the research that they've been doing here within the that's been supported by the Institute, but also by what's going on within the Epilepsy Research Institute. So if we can all just give a round of applause for everyone that's been going on here. So I just want to say that the last couple of minutes here is just to make sure that you can come and see us at stand 33 to chat to us more detail about more of the research that's going on within the Institute, and also to find a couple of points is to make sure that you join the hub so that you can keep up to date with all the other research that's going on within the Institute as well. And finally, just to make sure that you are, if you come and chat to us as well regarding our conference as well, it's going to be going on the 22nd and the 23rd of May. If you want to make sure that you can try, and we still got tickets for this, so please do come and join us at this conference if you're still able to join at this particular point. So again, thank you very much for joining us today, and I think we can all agree that it was some really, really amazing early career researchers that we're going on here. So we are radically advancing the research into epilepsy. So thank you again.